Welcome back. Do you have any questions with respect to the previous part of the session that we had on synthetic biology? If not, we will get into the enzymes and the engineering enzymes, and we'll also discuss about systems biology. The thing would be that some of the concepts we have already discussed while we were having the conversation about synthetic biology, so it might seem like a repetition. And we are also going to go through some of the details about enzymes that you might be aware of. If in case there is something that you would like to add, please let me know. We can add to our conversation, and if there is something that I am able to add to you, that would be perfect. Okay, to start with, enzymes. Now, what are enzymes and what is a catalyst? Catalyst, if I remember correctly from my chemistry, is anything that basically moves the chemical reaction forward in a specific direction. So, if you are moving from A to B, you use a catalyst, it will make it faster. But catalysts are always materials that will provide their surface for the reaction. Is this statement true? Majority of times, catalysts are the ones that are providing the surface for the reaction to take place. There are also ones which impact let's say, the activation energy that is involved in the reaction process. Now, all enzymes are proteins, but not all proteins are enzymes. That's, if I remember correctly, that was there in the biochemistry textbook. Now, proteins that speed up reactions in living cells to perform various functions like digestion, muscles building, and many more are one of the few of the functions of the enzymes that we are going to work with. The one which you see on the right hand side is a lysozyme. You should have heard about egg white lysozyme protein. So, the egg white albumin, or the egg white of the the egg white contains lysozyme as one of the proteins. Where else can you find lysozyme? Yes, in your saliva, right? So, lysozyme is a very important enzyme, and it's a very common enzyme. Now, most of them are three-dimensional globular proteins. That means the tertiary and the quaternary structure. So, when we were discussing about the structure of the DNA, we talked about the ATGC, but we didn't talk about the structure of the protein, right? Now, proteins are made up of amino acids. So, if you have amino acid A, let's say I'm just going to use A, B, C, D, E for that. So, you have amino acid 1, 2, 3, 4, 5, 6, 7, 8. So, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Now, 16 amino acids, if they are arranged in a sequence, this becomes your primary structure, right? Once they start folding, they will give you the secondary structures. What are the different secondary structures that you will have? Alpha helices, beta sheets, parallel and anti-parallel. You will also have coils, turns. What else are there? These are the major secondary structures that you will have, right? So, all of these secondary structures. So, what defines a specific secondary structure? That this particular protein sequence is going to give you a secondary structure, let's say alpha helices or a beta sheet. What defines that? What defines a specific protein would be produced in a DNA? The sequence of the DNA, right? The sequence of your amino acid is what defines what sort of secondary structure you will have in a protein. Now, when you take all of these secondary structures and then these secondary structures fold further, they give you tertiary structure, and when multiple tertiary structures combine together, that gives you a quaternary structure, right? Now, all these structures of the proteins, when you look at from the very first slide, if you look at from the surface dimensions, so if you add surface to that protein, you will see that there are grooves. These grooves are mostly the locations where they are able to go for their lock and key mechanism. So, these are called your active sites. That might not be the active site in this particular lysozyme protein, it might be here. But I am just saying the structure of the protein will define where exactly your active site will lie and what the structure of your active site will look like. Now, most of can be obtained from plants. Enzymes can be obtained from plants, from animals and microorganisms through relatively simpler extraction processes. It has been used for centuries in the food production process, for cheese making, beer making, beer brewing, and dough leavening. What other applications of enzymes do you see in the food sector?
Bitterness reduction.
So, you are talking about the sensory. It's just adding the sensory value to it, right? Can protein add texture? What else is there? The gluten protein that you find in the bread,
Like
what does it provide?
Blood, like cross-linking, softening of the blood.
It provides a close linking and the network in which your starch is available, right? So, that also adds to the texture of the bread. So, proteins are capable of providing you the textures. They are also not only capable of catalyzing experimentation, reactions, but they are also capable of adding texture, the sensory values to the food products, and that's why we are using them like a lot in the food industry. From the pharmaceutical aspect, enzymes are capable of working as a drug. There is a medicine which you should be aware of, Unigyme. If you have an upset stomach, that's when they are given that Unigyme. So, it helps you to make your digestion go well, right. So, there are different types of applications of these enzymes, because the enzymes are the ones which are going to cleave the proteins into smaller bits, and so that your body can absorb the amino acids. Any idea why we get allergies to proteins then? If we already have the enzymes and if they are able to go and cut the specific protein into smaller bits, why do we get allergies? Use the mic to answer the question.
Autoimmunity. Like
so, you are saying that when we ingest the protein, our autoimmune or our immunity, our immunological response against it leads to allergy. That yes, I am aware of, but
yes, sir. Like even certain protein, even our body is considered as antibody. So antigen. So, for example, egg allergy. Certain people egg allergy. So, for example, in case of gluten intolerance, celiac disease are there. So, it's like immunity, like even that may create certain problem to those people, like swelling.
No, I know. So that would be an immunological response to the presence of the protein. My question, I might not be very clear in that. So my question is, what part of the protein initiates that immune response? Is it a specific amino acid that initiates the immune response?
Receptors, like
so proteins have receptors. The body has receptors, but my question is, what part of the protein elicits an immune response? You are the biotechnology, you should have a course in immunology, right? I had, that was long time back. Try. See, there is no right answer for most of the things, so just try your best. And let's say, okay, so you are aware of nut allergy. Many people do have nut allergies. In one of the research that my graduate student did, we figured out that, not my graduate student, actually a colleague of mine did. We figured out that if you boil peanut, the allergenicity of that protein is less. So, the immune response that might happen is less as compared to if you are roasting that peanut. So, roasted peanuts will elicit a heavier immune response as compared to the boiled peanuts. Why? Proteins do get denatured at higher temperature. Roasting is at a way higher temperature, because oil is at a very high temperature as compared to water. There is a change in the structure, right? So, similarly, you can actually engineer your enzymes by, because all proteins are not enzymes, but all enzymes are proteins. So, because they are proteins, I can change the structure of my enzymes. By modifying the structure of my enzymes, I can change their capabilities, I can change the way they will behave in a specific chemical reaction. Okay? That is what I wanted to bring our discussion back to, but it is important for you to understand the structure of the protein, to understand how the functionality of the protein is dependent on its structure. If you change the structure, let us say if you change the sequence of the amino acid, you will change its functionality. But let us say if you are not able to change the sequence, even if you modify the secondary structures, you will modify the functionality of the protein. Okay? So, if I take a protein and let us say I put it inside a microwave and I switch on the microwave for a minute, because of the electro oscillating microwave, right, also oscillating electric field, my protein will go through push and pull, because your alpha helical structures are like a dipole. So, they have a positive and a negative. So, they will start rotating in the direction of the electric field, and that leads to the turns and the twists, turns and the twists. So, if you have like it's like a wire, so you keep on turning, twisting, you will break it. If you use enough power, you might be able to denature the whole protein. So, that what happens when you are working with the proteins with the temperature perspective. So, we do want sometimes for our enzymes to have the capability that they are not impacted by the temperature. They should not be impacted by the pH, because all these environmental factors, if they are impacting our enzymes functionality, we would like them to be safer. We would like to make sure that we engineer them in such a way that they are not impacted by these environmental factors at all. That comes under the engineering enzymes. Now, the structure of the enzymes is a globular structure with a pocket which is called as an active site. We already discussed that. Now, active site is where the binding of actives so with the residue takes place, and which help to hold to the substrate. And the active site is generally less than 5% of total surface area. So, if the design or the structure of the lysozyme that we saw, even just one single group might be the active site, rest all is not involved. It is only one section of the protein which is just maybe the 5 to 6% of it, of the surface area, which should be involved in the actively involved in the reaction process. So, change in the shape of the protein affects the shape of the active site and the enzyme function. So, if you take your enzyme, put it inside a microwave, take your enzyme, put it in the boiling water, take your enzyme, put it in an oven. The moment you will add heat, the protein structure will change, the active site structure will change. So, if you change the active site structure, it might not be able to bind. So, those are the things that happen. So, if there is a mutation that takes place and your enzymes are not capable of binding to a specific substrate anymore, then that particular mutation is working against you. If you come up with a mutation that is going to give you an enzyme with an active site structure that will actually work in your favor, is where I'm going with the directed evolution. So, if you want to change, we would like to have those type of changes to change the structure of the enzyme itself. And you can only change the structure of the enzyme by changing the sequence or subjecting it to external stress like heat, pressure, or even shear, mechanical shear, that might help change the structure. But those things are outside the scope of our conversation. So, we would only like to work towards the changing the primary sequence. That primary sequence change will give us a different change in the structure of the enzyme. Now, these are some of the classes of enzymes. You have oxidoreductase, transferases, hydrolases, lysases, isomerases, ligases. So, they all have different functionality. So, oxidoreductase will go for the redox reactions, transferases for transfer exchange of certain chemical groups, lysases would be on non-hydrolytic bond cleavage, Isomerism would be conversion of isomers from cis to trans or trans to cis, if I remember my chemistry correctly. Ligases or synthesis would be synthesis of two molecular substrates into one molecular compound using ATP hydrolysis. Some of the properties of enzymes which are of importance for us would be their absolute specificity. Some enzymes, if they have a specificity to a specific substrate, we would like to maintain that, or we would like to develop that type of specificity in our engineered enzyme for that specific substrate, so that we can get the product that we want at the very end. The group specificity would be, if they are structurally related groups, it can act on both the structures. Optical specificity would be to work on the optical isomers, and the geometrical specificity would be specificity towards cis or trans forms like fumarases or interconversion of fumaric and malic acids. The other properties of the enzymes would be the colloidal nature of the enzymes, the catalytic nature, we have already discussed that. It can be affected by the temperature and pH. Now, these are the most important parameters that we are going to work with. Now, when we were discussing about precision fermentation, when we were discussing about cellular agriculture, or whenever we had the conversation about the fermentation as part of cellular agriculture or precision fermentation, we didn't talk in detail about the process optimization. We did discuss that the process optimization is required, but what are the process parameters in the fermentation that are involved? So, you have a fermenter. Let us say we are just going to draw a simple fermentation system. So, you have your impellers, you have your sparger, so which is your O2. What are the process parameters here? The speed and the rotation of the impeller would be there one process parameter. O2 concentration, let us say, would be another one. The temperature can be another one. So, I will have a temperature probe, I will have a pH probe, right. So, I can change the pH, I can change the temperature. Now, will the enzymes get impacted by the impeller speed? Just guess. Will enzymes get impacted by impeller speed? Maybe, but it is highly unlikely, but maybe, because impellers are not moving like regular fans, the ones which are running around here, but they are just pretty slow. It's mostly to maintain the homogeneous mixture of the medium, right, or to make sure that oxygen is diffusing throughout the medium. So, when we come into the design of the reactor, you will see when we are talking about the mass transfer, how we want the oxygen to be diffused throughout the reactor, and that is where the impeller speed, the design of the impeller comes into picture. The other aspect, the ones which we can control and one which changes as the product is formed or the substrate is consumed, is your temperature and pH. So, the temperature and pH can impact whether your reactions are going forward or it is stopping completely or it is going backward, right. So, enzymes being impacted by temperature and pH is not something new for the fermentation process if the enzymes are involved there. So, it is a very important process parameter, sorry, these two are very important process parameters, and we always like to see if the enzymes or the proteins that are involved in the fermentation process are actually capable of running at the process parameters that we have chosen. So, if I choose, let us say, a temperature range of 50 to 80 degrees Celsius, my enzymes and the proteins that I am working with, or even my product, should be stable at this particular temperature. My raw materials should be stable, my product should be stable, and the whole process, the chemical reaction that is taking place, should be stable at this temperature range. pH mostly would be in a fermentation process. What is the general pH? Is acidic, alkaline, or neutral? Acidic. So, you are going less than 7. How much less than 7 do you think you go? Like 3, 4? It goes 6.5, 6.6, that is still neutral. So, you maintain somewhere near the neutrality. So, you want to maintain that. So, some of the products that are formed will add, like if ethanol is a product that is formed, it is going to increase your acidity or pH or decrease it? It will increase the acidity, right? So that means it will lower the pH. And when it lowers the pH, what happens to the microbes? They die, right? So that's why your substrate now is acting as a inhibitor. See, you all are aware of the design of reactors. I don't need to teach that now. So that is exactly what happens when you are looking at the substrate being produced, the product being produced, and sometimes the product will act as an inhibitor for the growth of the organism. Now that's where the enzymes also come into picture. So if I am able to maintain the pH by producing other products to counterbalance it, so there are things that can be done. Now it also speeds up the reaction by lowering the activation energy, and it should not impact your end product, the one which you are working with. Now the synthesis of enzymes, as we were discussing in synthetic biology, you have your DNA. So DNA gives you RNA or mRNA? And this mRNA gives you proteins, right? If I remember correctly, it used to be like one sequence is read, then another, there is a gap, and then another sequence that is read, and then that gives you the mRNA. Am I correct? It has been a long time that I have refreshed my molecular biology. But anyway, so you have your DNA sequence, you get your mRNA, from the mRNA you are getting your proteins, right? Now this is where your ribosomes come into picture, right? Now the information is carried by the DNA, which is clear, and the amino acid forms to make the specific enzyme. So the sequence of the amino acids that we are going to work with will define the type of enzyme that we are going to have. Now there are intracellular enzymes and extracellular enzymes. So intracellular enzymes are synthesized and retained within the cell. The example of those would be the reduction reaction in mitochondria. So some of the reductases that are generated there. Extracellular enzymes are synthesized in the cell, but secreted to work outside. Now when they are working outside the cell or outside in the cytoplasm, where? So when we say extracellular, is it outside the cell? Always. Intracellular can be in the cytoplasm, but it's still within the cell. Okay? So examples would be the digestive enzymes by pancreas to work in duodenum. Now many of these extracellular enzymes are also used to digest some of the food molecules that can be used by the cells for the growth, for their food. And you would be amazed to know that the capability of cells to produce enzymes, extracellular enzymes, is what make them very, very interesting. Like specifically microbial, some of the microbes, they can grow on plastic. They can actually eat plastic. There are fungi that can eat plastic. How are they able to digest plastic? It's not like they're eating like the way we eat. What are they doing? So they are producing enzymes that are basically breaking down the plastic into smaller units that can be absorbed and used as a source for the growth of the fungi or the bacteria. So that's why enzymes play a very important role if you are looking at from the environmental application also, not just from the food application, but from the environmental application. These enzymes play a very vital role. So currently there is a huge amount of research that is going on in this direction, where they are looking at identifying enzymes that are produced by different novel bacteria that can actually eat plastic. In my own research group, we are working on microplastics. So we are trying to identify microbes, or we are trying to identify processes in which we can actually remove these microplastics from the environment or from the aqueous environment. So understanding how enzymes can work or how we can engineer these enzymes might help us in future in developing products. Let's say we would be able to develop a product through fermentation, just like a dried enzyme powder. and you see that there is a water body, you know that it is contaminated with microplastics, you just spread that powder on that water body, and that particular microplastic would be degraded over a period of time. You should, do you have pets at home, if I may ask? So people do have pets, like specifically I have 2 cats at home, and pets sometimes, due to their behavioral issues, they will pee outside on your rug or on the floor, and that smell which is there, to get rid of that smell, there are products that have been developed naturally by using enzymes that will break down that urine and the odor completely. So that's a biological way of getting rid of the odor. That's the biological way of developing a cleaning agent. You would have heard about these enzyme based detergents, Oxy, which is used, Ariel, it has. So they also have these enzymes that actually work with the fiber to get rid of the dust. Where are those enzymes coming from? Where are those enzymes produced? In bulk. You can actually produce them through fermentation process or through chemically, but that's where one of the applications, the common application that you can see. So the mechanism of enzymes, that is basically your Fisher lock and key model, which is the most common that we are aware of. So it was proposed in 1898 by Emil Fisher. So you have the active site which is rigid, and there is no change in the active site. So the active site, if it is an L shape, it will remain L shaped. You cannot change it. So only substrate which are L shaped, which can fit into that active site, will be able to come and bind to the enzymes. So substrate is the key that fits the lock of the enzyme, and that's when the chemical reaction will move forward. Similarly, there is another one which is Koshland's induced fit model. Now this is where you are basically forcing the active site to take the shape of your substrate. Active site might be of different shape and size, but it will take the shape of the substrate. So it's flexible. So which one would be better, this one or the rigid one? For larger application, you want something which is much more flexible. For specific application, you want something which is very, very specific, right? I will just give you that as a hint. This will come in your case study. Think about it later on. Now, enzyme engineering, why do we need that? Why do I need to modify enzymes? We need to modify enzymes for the industrial and other applications. So, designing enzymes by changing the amino acid sequences through recombinant DNA technology, that is where your directed evolutions comes into picture, is one way of modifying it. Now, enzymes exist in nature, are taken and modified with random mutagenesis. Sorry, this is directed mutagenesis, sorry, evolution, the other one is not. My mistake, please. And you start with the protein from the public database, which is your PDB. You focus on its active sites, and you place the reagent or the substrate in a way to catalyze the reaction. Now, you would have heard in the bioinformatics about drug delivery, or no, drug discovery. And this drug discovery is through drug design. In bioinformatics, they do that a lot. What do they do it? What do they do there? Sorry. So, are they designing drugs that will fit the enzyme, or they are designing the enzyme that will fit the drug? Think. Once you are ready for your answer, you can raise your hand, we will give you the mic, and then you can answer that. So, are you designing your enzymes to fit the drug, or you are designing the drug to fit the enzyme? You would have heard this term, right?
Sir, yeah. So actually we are designing the enzyme because there is two kinds of docking when you do it, like rigid docking and flexible docking.
Yes.
So when it comes to flexible docking, we particularly select a particular like of the protein structure. We select a particular region where it's binding. So when you take rigid docking, it's take a like a huge section of the enzyme. But in this one's flexible docking, we take a specific section, and so we decrease the area which to where that specific chemical compound, which will later on serve as a drug, is binding to the enzyme.
So your enzyme structure remains the same, or you're changing the enzyme structure? And if you are changing the enzyme structure in your docking process, how are you doing that? Are you using different types of enzymes, or you're randomly deciding, you know what, it will look really good if it looks like an S, or it looks like an A or a B? How do you do that?
So basically, during the first, we then go for blind docking.
Okay.
Blind docking, it like we'll see which particular section or which chain is the chemical compound is binding.
Yes.
Then we select that particular section. We decrease the area to increase the efficiency where it's actually binding. So I believe it's it changing the structure of the enzyme. So it's to increase.
You're trying to increase the specificity.
Specificity, yeah. We're
right?
trying to increase.
But before that, you have your enzyme, you have your drug, multiple drugs.
Yes.
You do a blind docking, right?
Yes, sir.
Whichever fits.
Yes, sir. Whichever fits.
And then you start looking at studying the active sites,
active
right?
site, yeah.
That is part of engineering enzymes.
Yes, sir.
So your bioinformatics that you are studying right now is also synthetic biology. It's also part of your engineering enzymes. I just wanted to bring that. So because you are undergrad students of biotechnology, some of the graduate students here, so you should be, if you are able to go back to that particular course, refresh some of those parts, you will get a much better idea. Okay, so why should I modify enzymes? We would need to modify enzymes to produce enzymes for industrial and other application that was already there. And the other part is to improve the stability and the activity of the enzymes. As he was discussing about the drug discovery aspect, if you are changing the structure of the enzymes, you might be able to bind it to more drug. There are some specific drugs that will work. If you have multiple components that can work against your, or sorry, multiple drugs that can work against a specific disease, if you can make the modification in such a way that all those drugs can work, then the cost is less. It's not just one drug is available in the market. You can have multiple options, right? So increase operating time of enzymes and the duration to minimize the cost. The other aspect of engineerable enzyme would be the properties. What are the things that we are basically modifying, or what are the things that we are trying to modify? So you have your the pH range under which they will act, the thermal stability of it, the solvent tolerance. These are all the operational parameters, and the substrate product tolerance. Now these things will get used when we are working with the fermentation process, and we are using these enzymes in the fermentation process. And if you're looking at from the reaction perspective itself, you're looking at the different substrate ranges that it can work with, the specificity of the enzyme to a specific substrate, the novel reactivities like the reactions that it can generate, the cofactor use, and the kinetic properties, how quickly it can run that specific reaction, or how quickly it can catalyze that specific reaction. The other would be the immobilization and co-location. Now, this is basically based on where exactly you are locating it. I will not go into that. That is mostly on the structural basis, how easy is the access to the active sites. So if you want to make your enzymes much more accessible, you can engineer them in such a way that active sites are outward most of the time, so that you can bind to the substrate much more effectively and much more specifically. So that will improve the overall reaction process. Okay? What will be the easiest way to do that? If I give you an enzyme, just for our conversation, so let's say if I give you an enzyme, and those enzymes will have the polar surface area, non-polar surface areas, right? and the active sites will have the polar sites and the non-polar sites, because not all molecules that are on the surface of the protein are polar in nature. Some of them would be non-polar in nature. So, when you look at the very first diagram that we had, this one. So, let us say all the whites are the polars, all the blues are the non-polar. So, you have sites, different sites. It might be the other way around, but you have sites. So, you have your enzymes with the polar and the non-polar part. How do I change it? Like what will I do to its environment that it might change its surface structure itself? What can I do to the enzyme? What is the inherent property of a biological system? A biological system always wants to be in the most stable state possible, right? Structurally. So, if there is a balance between the polar and the non-polar sites for us, if I change the pH of the surrounding, the protein will try to rearrange its structure in such a way that it is reaching that balance again, right. If I change the temperature and the pH, I am forcing it to change faster maybe, but changing the environment, the pH specifically, because the isoelectric points are involved there, right. If I change that, it might change the structure of the active sites. So, that is why optimizing the pH, the temperature during a fermentation process or any biochemical process is very important, because if you are not able to maintain the conditions that are required for a reaction to occur, you are going to lose producing the product that of or the product of interest. So, that is why, because when we are working with proteins, when we are working with enzymes, things become a bit more complex. You want things to be very, very rigid in terms of the temperature range or the pH range. But if you are able to engineer these components in such a way that they are able to take care of their self for a specific range of temperature or specific range of pH, there is no modification in their structure, that would be an amazing opportunity, because that will reduce the rigidity of the process. So, the process might vary a little bit up and down, but you still get the product of your interest. You are getting the point what I am trying to explain, right. If it was very rigid, like if my enzyme is only is only active, let us say at a pH 7, what if my pH reduces to 6.8? Will it be active anymore? It won't. But what if it works between the pH of 6.5 and 7.5? It will be, because it will not change itself, it will not change its structure within that range, it will change the structure if it goes out of those range. That is what we want through engineering enzymes. We want it to be much more flexible. This is my understanding of engineering enzymes. If you read literature, you might get something else, but this is my understanding of it. Now, the engineering enzyme approaches that we have here. So, you have your engineered enzymes, let us say, which is 20x increase in the half life at 70 degrees Celsius. So, you are working with lipases. So, you do the solvent optimization, you do a substrate product tolerance of it, you improve the stability of it, you do a site directed mutagenesis, and then you do a computational design of it. This whole approach is part of your engineering enzyme or enzyme engineering. We are using the same synthetic biology tools to make modifications towards enzymes. If we go back, I might change the slides here. Let me go back very, very, very quickly. When we were discussing about the biomolecular engineering, the example that you see here is of new random mutations are introduced in the genes for selected enzymes. The cycle begins. So, you have your DNA, you do a random mutations in it. So, this is your directed mutagenesis, like you are doing a directed evolution. So now all these random mutations might lead to formation of different enzymes, like same enzymes with different characteristics, right? And then you would be able to identify these mutations. Now you keep on doing that, and then you see the changed enzymes are tested. Those that are more efficient at catalyzing a specific desired chemical reaction are the one, or the mutations that are involved in that. So what you have done is, like, you have, it's not just one DNA that you made the change, you have multiple DNAs, right? So for one you did this, for another one you did this, for one you did this. You kept on making different types of changes and you let it grow. They produce the enzymes with the modification. Now that modification, you test it against what you wanted to use those enzymes for. Whichever one worked, you go back and see which mutation actually led to that. You take that strain and then you continue. It's a very simple process, right? If you see it from this perspective, it's a very simple process, but it's a very effective one. It's long, but it's also very effective. Okay, so directed evolution, mostly for enzymes and proteins. For your case study 2, you have to think about directed evolution. C'est bon? Because I don't want you to blame me later on that I didn't help. So I'm helping you out little bit by little bit. So you have your case study 1 idea. We will discuss about that. I think I have already talked about that in synthetic biology. Case study 2 comes in engineering enzymes most of the times. Sorry, not most of the times, for this year. Now let's get into the established techniques of engineering enzymes. We have already discussed this. This is your directed evolution, which we talked about. De novo design is brand new design. You take the sequence, you design your own enzymes based on what you want it to work with. This is costly, but sometimes important when you are trying to develop drugs, specific drugs. It's a very costly process, but it's an important process. But you have to do multiple validations. It has to be go through. So you have your active site modeling. You change the sequences of the enzymes itself, and then you have the semi-rational mutagenesis, where your random and site-directed mutagenesis are connected. You have your sequence of interest in there, and then rational mutagenesis, and then you have directed evolution. This is, in my view, the most common one and the most effective one, the directed evolution. Now, enzymatic degradation of various types of plastics. This is a paper, or the work done by Choi et al. In 2024. This is a very recent paper. I will show you the reference, or you have the reference list in this particular slide deck at the very end. So if you get a chance, do download the paper and read about it if you are interested in microplastics. So different microbes will generate enzymes which would be breaking these microplastics. Now the point is, can I change the type of enzymes a particular microbe will produce by changing the feedstock that it is feeding on? It's just a question. I'm honestly, even I am thinking about the answer right now while I'm asking the question. But is it possible? Do you think it is possible to change the type, or to generate a specific enzyme by changing the feedstock? Because in my understanding, not the whole DNA is expressed at a given time. Depending upon what sort of feedstock I am having, my DNA might get expressed, and that expressed DNA might develop a different type of enzyme. Do you agree with this idea? You will see some microbes thriving with this change in the source, the carbon source that they get for their growth, and that's how they are capable of working or digesting the microplastics. They are digesting the plastics. These things these are some other sequence in the DNA that gets activated. This is my understanding of it, which was hidden, which was not active earlier, but suddenly it got activated because the source of the carbon that they had is not there anymore. So, they have to somehow adapt their whole biochemical machinery to take into account this carbon source, which they can eat and survive. And mutations move faster, right? So, if one progeny was not able to survive, some mutation would have taken place for the survival of the second one, the third one, and the fourth one, and the fifth one, and keep on going. That mutation will keep on going, and then you'll end up with a strain that would be capable of digesting microplastics completely. So, that research is very, very fascinating on microplastics. So, now this microorganisms are inhibiting various surfaces of microplastics, and they release various types of extracellular enzymes. So, you can see the list of enzymes here, which are able to degrade these microplastics into smaller fragments. So, PETase is the one which is going to digest the PET, and then you have the lipases, the proteases, lacases, cutinases. So, there are a lot of enzymes which are produced from different types of the materials. So, for the PLAs, PSs, for the PETs, these are the enzymes. So, which one can work on which type of biopolymer? Now, enzyme mediated microplastic degradation would also involve protein engineering strategies, which are employed in the modification of enzymes that can help in degradation of plastics. So, if you are able to make modifications to enzymes in such a way that you can develop it, that develop the active sites that can digest the microplastics, that would be one way of interacting with enzyme engineering. Now comes this idea of synthetic biology, the systems biology, and the enzyme engineering. It's a very interdisciplinary area, just like your biotechnology. It's a very interdisciplinary area. So, understanding of systems biology, synthetic biology, some of the computational aspects where you are using Python, which can use into bioinformatics, using BLAST, FASTA, and learning the DNA sequence technologies. All of this will give you the main, which encompasses everything. This is why I said understanding synthetic biology will help you pretty much understand enzymes, enzyme engineering, and systems biology. Now, it comes the systems biology part. So, holistic and dynamic interactions within the biological systems is required. So, the whole is greater than the sum of its part. So, what it means is that understanding the whole of an E. coli, let us say I do not want to understand just one pathway of the E. coli, I would like to study the whole metabolic pathway of the E. coli. What are the pathways, different pathways that are there, and how they are mapped. If I know how it is mapped, I would be able to engineer it in a much better way for my own benefit. It's like jack of all trade, master of none, but you really want to become master of one in this case. So, you want to understand everything. You do not want to be like just little bit of this pathway, that pathway. You want to understand the whole map of E. coli, how each and every pathways are interrelated, what product comes out, what product does not come out. And once you have that information, you would be able to engineer it for your own usage. So, that comes under system biology. So, system biology is an interdisciplinary science that studies the complex interactions and the collective behavior of a cell or an organism. So, your genes will basically give you your molecular networks, the molecular networks give out your cellular networks, and the cellular networks gives out your organ networks. Now, genes will lead to phenotypes, right, but those phenotypes, that whole process to reach that phenotypic effect, you have a metabolic pathway in between, right? And it's not just one gene that is involved in that particular phenotype. There are multiple genes that would be involved into giving that specific phenotype to you. So, understanding the interaction between those genes, understanding the interaction between the metabolic pathways of those genes that led to that phenotypic change is part of the systems biology. So, this would be an example. So, you have an existing drug. So, let us say if there is a patient, that patient requires that drug to get rid of a specific disease. So, you give that drug to the patient, whether, and then you get a thumbs up or thumbs down, whether it worked or it does not work, right. So, that is a straightforward way of doing it. But before any drug is launched, we always try to understand its efficacy. You remember the term that I used yesterday, right? The Dolo 650. Why 650? Why 650 milligrams? Because that 650 milligrams is more than enough to reduce your fever. That is why we cannot say how many glasses of orange juice you have to drink to be cancer free, because we do not know the dosage value. So, dosage is very easy to give for the chemicals, because we can develop that efficacy. So, but that comes out only from clinical trials. That dosage value comes out from the clinical trials. Even your COVID-19 vaccines, the one which we got in India, I think that was AstraZeneca. Covishield, that was developed by AstraZeneca, or AstraZeneca, we will call them the same thing. And rest of the world, it was Pfizer and Moderna. Yeah. So, Moderna and Pfizer, they were mRNA based, right. Now, this mRNA based, that was an amazing application of synthetic biology in developing a vaccine. Now, think about it just for a moment. Just to develop a single vaccine, you have heard about flu shots, like people get a flu shot, like flu vaccine. Like in North America, we always get, like every flu season, there is a vaccine. But that particular vaccine is good for the flu or the whatever the host was last year, not this year. So, I got the flu shot before I came to India, but I got the flu shot for the flu that was last year. This year one comes next year. So, it's a long process, because they have to sequence it, identify, develop the vaccine, and then only then you get it, right. But how was this COVID vaccine produced so quickly? It happened, and within a few months the vaccine was available. That is why we were able to save so many people, right. It was all because of the understanding that we have had through systems biology, the synthetic biology. Because once we knew how the whole pathways work, we were able to do the genetic circuits, take out the sequence which was impacting the active sites related, develop the vaccine and make it ready for the market. It was so quick, but it took a lot of time. But you do need that clinical trial aspect to it, right. That is where existing drugs come into play. So, where you have to go for the clinical trial. So, you have your molecular biology, you do the clinical trial, and that you develop a mathematical model using the systems biology thing, and then you can have this personalized drug which would work for the patient, because not everything will work for the patient. I will give you an example to it. So, rosuvastatin or rosuvastatin, or R, is much more easier. So, R, which is a cholesterol lowering drug, its interaction is consistent. It's a very consistent drug from one human to another human. It has nothing to do with your what is that called, variabilities. So, like the model helped researchers understand why the drug works consistently despite genetic variability. It will keep on changing. Now, it was mostly physiology, like you have these two terms like pharmacokinetics and pharmacodynamic model. Both of these models were used to develop this particular drug. So, how the, like once you take the drug, how does it travel within your body? That was understood. What is the site at which it will go and work? That was understood. Once it reached the site, what does it do? That was understood. Once you have all of that information, you can develop a drug that would be consistent for me, consistent for you, consistent for the other. It will work in every human person with the same efficacy. That understanding comes from understanding of the systems biology. I don't want you to like remember this, I am just giving this as an example. This is an application of systems biology. The other comes the omics and the cascades of the omics. For understanding your system biology, because you need to understand each and every metabolic pathways and the proteins that are being generated by each and every gene, the RNAs, you have to go for epigenomics, you have to go for genomics, transcriptomics, proteomics, metabolomics, cellulomics, and the single cellulomics. Once you have all of this information, you can make maps or networks. So, you will have this particular, so let us say this is a genetic sequence, this is giving this is giving to another pathways, this will lead to the other set of reactions and so on. So, you will be able to make a genetic map or metabolic map of the whole sequence. And this particular cascade is basically your flow of information through the biological systems. It gives you the hierarchy of how the things go through. So, you have your histones, modification, DNA, methylations, 3D organization of chromatin that goes into DNA, the DNA gives you RNA, RNA gives you protein, protein gives you metabolites, metabolites gives you cell functions and the phenotypes, and then you are getting back. So, synthetic biology and systems biology have a synergistic relationship. So, systems biology is understanding integration and prediction using computational models. So, once you have all that information using all the omics, you can develop mathematical models. Synthetic biology is control design and engineering. Using computational model, you are designing new things, specific molecular events. So, you combine both of them, you get where the system and the synthetic biologies are used in synergy to have a desired system behavior. So, if you want your system to act in a special way or specific way, use both systems biology and synthetic biology, understanding of both the sector, and then you would be able to get what you want. Most commonly for therapeutics and drug discovery, it is very necessary that you have these understandings. So, if you have a background in molecular biology, if you have an interest in molecular biology, cellular biology, develop more of that interest in computational biology, look into systems biology, look into synthetic biology, use that, and bioinformatics for sure for drug discovery. So, these are the references. The Choi et al. Should be somewhere here. If not, I will find it. Yeah. So, this one is the paper. So, recent advances in microbial and enzymatic engineering for biodegradation of micro and nanoplastics. So, this is one of the papers that my student was following for his research, and he helped me develop the slides for this particular session. We will take a break for 15 minutes and then we will get back for your case studies. We will have a brief discussion about it, because we have a complete session on this case study 1 and case study 2 later. But I want you to understand what they are, what we are asking for, and it will also require you to work in groups. So, if you can divide yourself into groups depending upon your own comfort zone. I am not aware who is comfortable with whom. So, if you can divide yourself into groups, that would be easier, because you would be working, let us say, on like sort of like a term paper at the very end, but it's not, I am not asking you to write me a 50-page or 60-page term paper. I would like you to develop the whole process for me. So, if this is the product that I want, this was the approach that I followed. So, I first went here, identified the sequence, I did this, this is the host that I selected, why I selected, what was the criteria that was based on it, what I did after that, and how I did it, and what sort of fermentation system will I choose to grow this particular product, and what would be the process parameters. So, you might have to do some Google searches, but it would be an interesting experience, because this is exactly how we develop research projects as researchers. We only know the product, now we have to develop the whole experimentation. Siva? Cool. So, we will take a 15 minutes break and then we will get back here.